Well, we are a little behind on quizzes. So I got 7 up, and we'll try to get 8 up um, early next week, and then we'll move on to Chapter 9's quiz. I got my highlighter to work here, so I'll be putting up those PowerPoints from Chapter 9 with the highlights. But we'll kind of go ahead and start continuing on with Chapter 9, talking about genetics and propagation, and moving on to the propagation part. So a little bit of review. Whenever we have cultivated varieties that originate from one single plant, any kind of asexual propagation, we're creating clones. We're creating genetically identical plants to the parents. And basically that is through kind of the asexual reproduction of the cells, mitosis, rather than through the sexual reproduction, meiosis. Many different ways, and we'll talk about some of those different ways of vegetative propagation. Looking at sexual propagation, though, if we are trying to maintain a cultivated variety, which, just to be specific here, cultivated variety being kind of a step further than just a species. Species of plants can basically cross-pollinate, reproduce, and retain similar characteristics. Cultivars can do that as well, but it's a little bit more specific, and things can vary quite a bit in the species level. But the cultivated variety aspect, we're looking at very specific traits, like in heirloom tomatoes, for instance, color, flavor, shape, and size of the fruit. Of course, there are other genes there that are important to look at in terms of what climates they grow well in, how well adapted they are to drought stress, things like that. If we have a homozygous cultivar, which again, homozygous means that basically they have the same alleles for a trait. If that cultivar cross-pollinates with other cultivars or even other species, then the resulting seed will be different than the parents. So we have to separate them from other plants and keep homozygous cultivars together in order to maintain genetic purity. And this separation, when we're talking about seed saving and breeding, we refer to that as isolation. And there are many ways to do that. You can isolate things in space, and generally for most varieties out there, or most species, I should say, most crop species, there are isolation distances that are listed for those crops. And even if it's a self-pollinating crop, you can have cross-pollination, especially if we have certain types of pollinators. For instance, bumblebees, they're great pollinators, but they tend to sometimes bypass some of the regular pathways. Sometimes bumblebees, for instance, will actually open up flowers by eating the outside of the flower and then getting to the nectar or the pollen without actually causing self-pollination. So you get a little cross-pollination there in certain cases. So things like peppers, for instance, they are self-pollinating, but they say that they should isolate peppers from different varieties by about 20 feet because of the potential for some incidental cross-pollination. Of course, in quite a few cases where plants are propagated by seed sexually, you want some variability. You want a little bit of heterozygosity, because that allows for more adaptation in the crop. Let's talk about seed a little bit. Many states and countries have their own seed certification programs, basically that are put in to kind of maintain genetic purity and the quality of seeds of different cultivated varieties. And these are kind of looked at by government agencies usually. Sometimes we have third parties doing seed certification as well. In the U.S., we have seed certification programs for 4 classes in our agronomic, our kind of commodified crops. We have breeder seed that are controlled by the breeder itself, and those are really small amounts and just used for breeding purposes. Foundation seed are those that are multiplied from the breeder seed and then available in limited amounts, often controlled by particular organizations to maintain seed stocks. Registered seed is for growers of certified seed, and they have strict guidelines of how to grow the seed, how to dry it, etc. And then certified seed is looking at the genetic identity and purity, and usually that is what is sold to farmers for crop production. Seeds have 3 essential parts. The embryo, kind of the center of the seed, which eventually will develop into the new plant. Then we have food storage material inside of the seeds, often in the form of carbohydrates and lipids. That is basically the food that gets the brand new embryo, feeds the embryo until it can photosynthesize and make its own food. And then we have seed coverings, and seed coats and coverings are there basically to protect the seed, protect the embryo, and keep it dormant. For a seed to germinate, of course, the embryo has to be alive, and in some cases, embryos can be very short-lived and only viable for a few days after they drop or months. All seeds reduce viability over time, and that's going to depend on your storage conditions. There are half-lives of seed listed out there for a lot of crop varieties that basically say how long it's going to take for that germination percentage to be reduced by half. And rarely do we ever have 100% germination. Have seen it happen before, but usually we're looking at 99% or below. And again, that germination is going to be different depending on different storage conditions. Ideal storage conditions would be in a sealed container under refrigeration at low relative humidities. There are cases out there, though, where we've found seeds to be viable for hundreds of years. They found lotus seeds, thousands of years actually old and still viable. They've also found bean seeds. There's a variety called the New Mexico New Mexico cave snap bean, and that bean was found sealed in a kind of a clay pot sealed with pine tar in a Native American settlement in New Mexico, and they carbon dated it and found the seeds to be about 800 years old, and some of them were still viable. So that's actually a variety you can still find today. So storage conditions maintain viability. It's a good idea to test the viability. We can do that through cut tests or X-ray tests in some cases, and probably the most practical and most common is the float test. I'm sure you guys have probably done this before, but basically you take seeds, drop them in water, kind of leave them for a little while, and if they float, those floaters are empty seeds. Often there's not an intact living embryo inside, and so they have air pockets in there which cause them to float. That's not the same for every seed, of course. Some seed is naturally dispersed by water, so they naturally float. So we don't That's not a surefire way, but for most crops, the float test does work pretty well. Of course, if you're doing the float test, you're actually encouraging germination by increasing moisture. So you really have to plant them soon after they're floated. We also have germination tests where basically we take samples of seed lots and separate them and just count them and see how many germinate. Of course, we do that in several different test lots and then average out the percentage to see what the variety is like. Often, if you're selling seed to seed companies, which there are a lot of, actually quite a few in Virginia, they will require you to do a germination test before you even send the seed in. There's also a tetrazolium test where we use this chemical to basically, when it touches respiring living tissue, it will turn red. And in order to do that, they will soak the seed, then cut it open to expose the embryo, place it in that tetrazolium solution, and then see if it's viable. Not super practical because, I mean, it's a way to kind of get an idea, but you have to actually cut it in half in order to be able to test that. And at that point, you're exposing and potentially damaging the embryo. However, there is an excised embryo test where you can actually remove the embryo and place it in a moist paper type of a specific type of paper in order to see viable embryos having some activity. You can also place that on an agar media, which we'll talk about here in a little bit when we get to tissue culture. Dormancy is, basically what seeds are. They're dormant plants. They're still alive, though. I mean, we have a living embryo in them. It's very important for many species to have dormancy, though. In order to preserve seed and for us to be able to take seed and save it for the next season, we have to have dormancy. There's different types of dormancy, which result in different structures of seeds, different physiological conditions, different shapes of embryos, and basically different ways that we can germinate them and actually get seeds to grow. First, we got seed coat dormancy, where actually the hard seed coat on the outside of the seed is naturally impermeable to waters and gases unless it is the proper environmental conditions for them to initiate germination. And we have several methods of removing the seed coat or softening it in order to allow for germination. Big one is scarification, and that is where that seed coat is actually mechanically altered or chemically altered. Really, we look at both physical and chemical means as scarification. So really, the next couple here, heat treatment and acid scarification, those are both just types of scarification that basically disrupts the seed coat and allows it to germinate. And these scarification methods do happen in nature, often through abrasion of seed coats from like freeze and thaw cycles, more rough particles in the soil. Also, the acid scarification is much like passing seeds through the digestive tract of a living organism. We also have stratification. It's important to note the difference between scarification and stratification. Stratification is a cool, moist treatment that allows seeds to germinate, and most of our native kind of temperate zone plants have to have some sort of stratification. So that can be emulated. You can either just kind of keep it outdoors in the winter as long as it's kept moist, or you can do things like stick it in a freezer or a refrigerator to induce stratification. We have to have chilling temperatures. There has to be moisture to maintain the integrity of the embryo. Also adequate oxygen, and of course time. So those are usually the factors that go into stratification and different periods for different types of plants. And they found recently that stratification actually will increase growth promoting hormones and reduce growth inhibiting hormones. So it'll promote things like the gibberellins and cytokinins and remove or inhibit abscisic acid to allow them to germinate. Some species have both seed coat and embryo dormancy, and that's called double dormancy. These tend to be more difficult to get to germinate. Sometimes it might even take several years or several seasons to get it to germinate. We don't really see this in any of our main crop species, but some kind of alternative crop species, we do see it in. We've been working with pawpaws, for instance, a kind of native sort of tropical-like fruit that has this really difficult double dormancy to overcome, and it can take over a year to get the seeds to germinate. Sometimes they need both scarification and stratification. Germination proceeds in different ways for different species. And here's just a few images kind of showing germination and how it occurs. And here we're kind of looking at more of like a fruit tree, in this case a peach. It's just a little bit different process of how the radicle emerges, etc. The environmental factors that influence germination are important. Of course, we have to have moisture, enough water to enter the seed coat and then trigger the embryo to come out of dormancy. Temperature, of course, and all of our crops have listed, you can find quite a few different resources about their different temperatures, the minimum, optimum, and maximum temperatures at which they will germinate. And it's often a pretty big range, but for the best germination and also the best vigor of seeds upon germination, what we really need is the optimum temperature. Aeration, we also have to have oxygen in a germination environment. It can't be too wet. If it's too wet, then that reduces oxygen in whatever medium you're trying to germinate seeds in, and the seeds will rot rather than germinate. Also, in some cases, they need light or the absence of light. Some seeds are, not as important for this, but things like lettuce, celery, parsley, a lot of grasses, they naturally shed their seeds and they lay on the surface of soils, so they actually need some light in order to germinate. A lot of flowers do that as well. Also, they need freedom from pathogenic organisms, especially damping off organisms, which are caused by these fungal and bacterial organisms like Pythium and Rhizoctonia. And finally, we need freedom from toxic amounts of salts. That's become an issue in areas where they have widespread irrigation and in kind of arid areas. Accumulation of salts in the soil can prevent germination. Let's get on into the vegetative, the asexual side, and all asexual reproduction is conducted through mitosis, the non-reductive cell division. So the genetics stay the same. And here's just kind of a picture sort of or illustration of meiotic cell division taking place in different parts of the plant, in the stem tip, in the root tip. Genetics stays the same. This meiotic cell division can produce adventitious shoots and roots, and often they need to produce a callus first in order to have successful asexual propagation. So here's adventitious shoots appearing, and these shoots are those that basically appear any place other than where they're expected to appear. at the terminus of the shoots or in the axils of the leaves. And adventitious shoots are often known as suckers when they're unwanted. We also have to have adventitious roots, and we need both roots and shoots. Often we need roots before we need shoots in order for plants to have successful vegetative reproduction. Adventitious roots are the same. They appear from any place other than where they're expected. They can come out of the stem, for instance. We were looking at that in corn, for instance. They create these brace roots, which are natural adventitious roots that help to keep the plant standing up. Generally, we have to, in many species, not all, but in many cases, we need a callus, which is basically just undifferentiated parenchyma cells in order for roots to be able to develop. Vegetative propagation is mostly used for highly heterozygous plants, those that do not breed true to type from seed. So in order to maintain the genetic identity, we basically clone them using vegetative propagation techniques. In some cases, we use grafting, which involves the use of a scion, which is the top part, and then the rootstock, which is, of course, the root system. And we'll get into that a little bit here when we talk about grafting in a little bit more detail. Almost all of our fruit and nut cultivars and many of our ornamentals are clones. They have that same genetic makeup, the same genotype, and potentially can all be exactly alike. That depends on the phenotype of the environment and how it causes those genes to be displayed. We can create cultivated varieties in kind of two different ways, cultivated clones as seedling plants propagated vegetatively, or also from mutations. Usually we call these mutations bud sports, and that's basically just where it starts with a single cell that is mutated, and then it could grow on even just one area of the plant. If you can propagate that particular area of the plant, that spore, then you can create a new cultivated variety. As I mentioned, the phenotype, the environment can cause the genetics to appear differently, even though they are basically genetically the same. Mutations can also occur even in vegetatively propagated material with the spore. Some mutations only change just a portion, and again, that's often known as a spore, but they are also called chimeras. And the issue of chimeras is fascinating because it can be difficult to try to propagate chimeras depending on where these chimeric mutations kind of appear. And this fruit right here, this variegated pink lemon, they call it the Eureka lemon. It's a variegated chimera, where basically they witnessed on one branch of one tree this mutation where they had variegated foliage and also variegated fruit. They were able to clone that and reproduce it, and now you can find it in nurseries across the country and around the world as well. So looking at the chimeric plants, kind of based on the location and the relative proportion of mutated versus non-mutated cells, we've got mericlinal, periclinal, and sectorial chimeras. And as you can see here, the periclinal kind of occurs up here in the apical dome, if you will, and often that spreads the mutated region throughout the newly growing portions of the plant. Sectorial and mericlinal areas are not in that apical dome, and therefore they only tend to occur in a very small portion and are more difficult to propagate. So again, just basically just repeating what I just said there. Periclinal chimeras are relatively stable because they're positioned near that apical dome, and every cell that reproduces from that will have that same characteristic, whereas the mericlinal and sectorial chimeras don't necessarily do that and are difficult and in some cases impossible to propagate. So where that chimera occurs is important to look at as well. Of course, we can have major issues with diseases, viruses that can occur in vegetatively propagated material. If you think about it, really, clones are more susceptible, and that is because they have the same genetics. They can't adapt to differences in environments, or they can't adapt to diseases because they are only asexually propagated. That's the whole reason why the Irish potato famine caused by the potato blight was such a big deal, because they were only growing one variety of potato. So they were all clones, and they basically wiped them all out. There are sources today where they actually register virus-free seedlings so that it's not transmitted in clones. We can also, and basically that's what they do, is they remove using heat treatment or chemical treatments to remove the viruses and other diseases from the clonal material. And we could potentially, let's just say that we have an apple variety that we want to reproduce, but it has fire blight. If you're able to obtain a cutting and then sterilize it, basically, then you can eliminate that disease and still get the variety. Another way to do this is through shoot tip culture, and we'll talk more about that in just a little bit. Let's talk about the different types of vegetative propagation. First, the most common and probably the easiest way to propagate most plants is through cuttings. And we can look at cuttings. There are different types of cuttings from different parts of the plant. We have stem cuttings, leaf cuttings, leaf bud cuttings, and root cuttings. And recognizing which plants do best under which types of cuttings can lead to more successful propagation. Here are some different types of cuttings just pictured here. On A, we've got a hardwood stem cutting, and there are other types of woody cuttings. Greenwood or softwood cuttings are kind of taken during the summer and during a flush of growth. Hardwood is taken when they're dormant after a flush of growth. B, there, we've got a leafy stem cutting. C, we've got a leaf cutting where basically we just took a leaf that was able to produce a shoot. On D, we've got a leaf and a bud. Those are usually more successful because you have both kind of leaf meristems as well as stem meristems there. And then E is a root cutting taken from a section of root. Let's talk about grafting for a second. And grafting and budding. Budding is really just a type of grafting where you're only using, one bud to graft. Basically, we're joining parts of two different plants together so that they unite and continue to grow as one plant. That, again, it involves the scion, which is the upper part that will eventually become the above ground part of the plant, and then the rootstock, which is the lower part that becomes the root system. We got root grafting, which basically we're putting a scion directly onto a very short piece of root. Top grafting or top working, we're often changing the cultivars of a fruit tree or a grapevine by removing the current top and then adding a new scion. We can also use grafting as a way to repair trunk damage in trees by doing things like bridge grafting, or to replace potentially an injured root system by using a technique known as in-arching. And those are really complicated, difficult techniques to do, but they can be used. Here's just example of probably the most common whip and tongue graft, which basically we make a cut, usually at like a 45-degree angle, and then make a little notch in that cut so that when you put the scion onto the rootstock, it will naturally be kind of held in place there in that notch. So you want to do the same, kind of the opposite on the scion as you would on the rootstock. The opposite cut, you want to try to do it at the same angle and try to make that notch appear just to where they line up perfectly. And this is what it looks like when you've done that, where these tongues interlock. And then you tie it and wax it or tape it. You know, today we actually have devices to do this. We have a grafting device which basically makes the perfect cut on both ends. So the big thing there is you just have to match up the diameters. You can do a smaller diameter of a scion onto a rootstock, but you cannot use a larger diameter. The big deal with grafting is that you have to match up the vascular cambium, which when you make a cutting for a scion or a rootstock, you can see that ring of vascular cambium. It's often the area that appears to be the most green. Looking at top grafting, this is a cleft grafting tool, and we've used this to top work different trees. Basically, you split the trunk of the tree using this tool and a hammer. You don't want to go too deep, of course, to get that cleft. You could use an axe even, but generally you want something that is not a very wide blade. I mean, you can even use a large knife or machete too. Then we, with the scion, basically, we have to remove the outside edges on each end so that we can get that vascular cambium to line up. And you have to do two pieces in this case. So we will insert the scions into the stock by first splitting that stock and then holding it open with a wedge, and then putting the scions in place. Kind of pictured here, putting those scions in. Notice they got that wedge shape on each of the scions stuck in there. Of course, you have to have the bud tips pointed up or it won't take. And then after you get it there, you wax over that area. The wax is meant to hold in moisture and also meant to prevent insects or disease from getting into that graft union. We've got budding as well, and T-budding is the most widely used form of budding. It's basically when you just take a bud or in some cases a piece of branch. We make this T-shaped incision into the bark and place that, the bark of the rootstock that is, and then place the scion in as well, and then tie it or wax it, tape it, tie it, tape it, wax it back. That union there will heal over by the callus production from the cambium layers of both the stalk and the scion. If you have it perfectly lined up, the cambium will act as one, basically, and continue to create new phloem and xylem that allows the plant to conduct moisture and nutrients. And this healing times, this union, often takes about 2 weeks before it really even begins to start. And during that time, we wax it in order for the scions not to dry out. We have to do grafting when the scion is dormant. You can do grafting and especially budding any time of the year as long as you have dormant scions. If the scions have already budded out, it's too late. And here's just a picture of that cleft graft. And you can kind of see up here where the bark lines up with the bark, and although it's not labeled here, this kind of layer right here would be the vascular cambium lining up with the vascular cambium. You got to do it on both sides, and they will basically unite with the cambium, and then it'll callus over that cambial region and cover that entire graft eventually. And here's just an image of that, kind of occurring, where basically you have the interlocking parenchyma cells, that callus tissue that seals the deal and makes it all work out. And then there we have new xylem and new phloem forming and allowing nutrients to be conducted as well as water. Whenever we look at grafting of any type, we have to have some degree of botanical relationship. And the closer they are related, the better. The more successful a graft it will be. Although there are some examples where you can do intergenera grafts, we don't know of any cases where you can do grafts in between different families. So at minimum, they have to be the same family. The closer the genetics, the better. Same genus is good, same species is best, the same genetics. I mean, if they're clones, then you can get, often 100% success rates. And for those of you that are interested in doing grafting, especially if you're into fruit or nut production, or even vineyard production, I would recommend starting off with clones, and just to get your grafting technique down. That's a good confidence booster. In some cases, we do have pear cultivars grafted onto quince roots. That's an intergeneric combination. Pears are pyrus, quince is a different genus, but you can actually get those to combine. Interestingly enough, though, it doesn't work the opposite direction, and we don't know why, but a quince on pear roots will not work as well. We've actually had some success here on campus grafting pear or apples onto these invasive calorie pears using the cleft grafting technique. It's pretty cool. If I can, I'll take a picture, put it up on Blackboard so you can see exactly what's going on there. And when you do that cleft grafting, eventually you usually want to remove one of the scions, if they both take, so that one of them becomes the new leader of the tree. There's examples of plums being grafted on peach roots, but peaches on plum roots usually don't work. Again, we don't know why. If you guys have ever seen in some nursery catalogs out there, they have like the fruit cocktail trees. It does work with plants in the same family, and they've had success. If you do a Google search on the tree of many fruits, it will come up. It's kind of interesting because it's actually an artist rather than a horticulturist that's doing this. But they've put almonds, peaches, plums, pears, and apples all on the same tree because they're all in the same family, the Rosaceae family. Generally, if you're looking at grafted plants, you can see the graft junction, like it's pictured here in this crab apple. You can see right there where that junction is occurring. There's a bit of swelling there, and often you can see that in plants for the rest of the plant's life. Big type of grafting in orchard production, pomology, is using dwarf rootstocks so that harvesting is easier. In some cases, we do herbaceous fruit and vegetable grafts as well, especially nowadays since methyl bromide has been banned as a soil sterilant because of environmental issues with it. Pretty interesting, though. We got lots of heirloom tomatoes now that they have a disease-resistant rootstock, but then the heirloom, the flavorful variety grafted on top. Moving on, another vegetative propagation technique is layering, and that's basically where the new plant actually roots while it's still attached to the mother plant. And it usually takes longer to do layering, but it's highly successful, really good rates of success in layering. We've been doing some in our plant propagation class here where we get like a 90% success because it's still attached to the mother plant. You're still getting moisture and nutrients from the mother, but it's also producing roots. So we got tip layering, and that's really only done in commercially in bramble fruits, the rubus, the raspberries, and blackberries, where basically you just bury the tip in a new adventitious bud, and then shoot forms. Then you just separate it from the mother plant. You got a new plant. Mound layering is often done in fruit trees, where basically they will cut a fruit tree back to pretty close to the ground to stubs, and then they'll cover those stubs, and then the new shoots will then also produce roots. Then you separate them from that. You can actually use that mother plant multiple times to do that same thing. Simple layering is basically just burying a portion of the plant. Often it's good to do some sort of wounding to encourage adventitious roots to form. And they're just talking about doing that there. Air layering is a little bit different because you don't bury it. Instead, you remove some tissue, often the bark from the outer layer of bark from the plant. Then you cover it with a medium, like often sphagnum moss, wrap it in plastic or really anything. We usually use transparent plastic so that you can actually see the roots forming. And then the roots will form here in the air layer. You can separate it and then later on plant it. You got a new plant. The most difficult thing here with air layering is ensuring that the medium remains moist. So often we found that we like to poke holes in the plastic so that you are actually, when you're watering that air layer too. Some plants, of course, naturally reproduce vegetatively, and they have specialized plant parts. In this case, strawberries create runners, and you can easily just snip those off once they develop some roots and replant them. Spider plants, same way. These are called stolons botanically. We often just call them runners, though. Natural plants. In some cases, we have adventitious roots being formed from, say, the horizontal root system, but actually, technically, it's the stem, a horizontal stem growing underground, a rhizome. And we can simply just divide these with a sharp tool like a spade or a shovel and just pull them apart. As long as they have a root, shoot, and bud, they will grow. Most perennial plants kind of naturally do this because of the crown naturally expands. And if you see like perennials, a lot of flowers, for instance, this crown as it expands throughout the years will cause the shoots to kind of flop over. That's a surefire sign that you need to propagate it in order to keep the health of the plant up and keep it flowering and keep it aesthetically pleasing. In this case, this is like a Shasta daisy here. You could separate each of those stems and leave the crown intact. You get new plants, and you can still get the existing plant from the existing crown. A lot of plants, especially herbaceous perennials, have their own unique structures that they naturally form, and you just have to separate them in order to propagate them. Things like bulbs, corms, tubers, tuberous roots, and rhizomes. We have separation, which is just the natural pulling off of, in the case of bulbs, they form little bulbils. In other cases, we have actual division. That's the mechanical separation, actually cutting the roots. Kind of skip over the types of bulbs there. We got corms. That's basically just the underground base of the stem is swollen, and then it has nodes and internodes. These are mostly just flowering plants that form corms, herbaceous kind of ornamentals. you guys grow potatoes, you're doing some type of division. You're doing some asexual propagation when you cut that tuber into sections where each section has a bud, an eye on it, and then you plant those and grow them. All potato cultivars are clones and are propagated by dividing each tuber into several sections. Often, the larger seed potato, you can get multiple plants from that one potato. Again, in other cases, we've got tuberous roots and root tissues that we can use to propagate vegetatively. One good example of that is, it's a little bit different than regular potatoes. Sweet potatoes, of course, botanically entirely different. We don't actually use the sections of the roots to create new plants. Instead, we use the slips or these adventitious shoots that appear. They're really easily just stuck in water and rooted. Here's just an example, a picture of a rhizome here in bamboo, where as long as you have a root and a bud, just even a very small section of rhizome will give rise to a new plant. In some cases, we have thick and fleshy rhizomes. Other cases, they're more quirky, more difficult to cut through, more woody. Some of our economic plants that are propagated from rhizomes include bananas, ferns, ginger, and many grasses. Bamboo is a grass too, it counts as them. The above ground horizontal stems are termed stolons, and they can be propagated the same as rhizomes. Basically, you just divide it, separate it from the natural plant part, and replant it, regrow it. Let's talk a little bit about micropropagation or tissue culture, where we basically take what are known as explants, which is just a piece of a plant. We grow that on a sterile nutrient media under aseptic, again, sterilized conditions. They're sealed. They don't have any outside environmental conditions to compete with. We're basically coddling the plants in this case in order to get them to produce. You can take an explant, though, technically from just one cell of the plant and reproduce it. So the good thing about tissue culture, although it's not super easy to do, is that you can produce tons and tons of plants from one piece of tissue. So what we're looking at is basically taking this excised piece of the plant, the explant, placing it on a media in order to, or even an embryo, placing it on a media, and then encouraging it to form calluses and grow. In some cases, they've even done that with pollen. Unfortunately, the nutrient media that we use for micropropagation is often also favorable, especially the environment that we promote too, for the growth of bacteria, fungi, and yeast. And for that reason, we have to sterilize everything that we use in tissue culture, not just the nutrient medium, but also the tools that you use, any scalpels or anything. You have to sterilize the containers that you use, so that you don't have any spores or any bacteria in there. It's something that is conducted usually in a lab environment. The nutrient media that we use to actually reproduce the plants on includes mineral salts, vitamins, sugars, growth regulators, and the organic complexes. Basically everything that a plant would be creating itself in photosynthesis, as well as nutrients that would come from the soil. We often get these from sources such as coconut milk or yeast extracts. Even banana puree is used to prepare these nutrient media. And they're often combined with a gelatinous, like agar type media, grown in containers. Right here, it's just kind of a, for particular types of tissue culture, the different ingredients they use in the nutrient medium. Once you have the nutrient medium, then you induce different types of growth by using different hormones. For instance, you'd use auxins in order to produce roots. You'd use cytokinins to produce shoots. You use those different hormones to trigger it to become a new plant. A huge step, though, in that, once you've actually got rooted plants, again, they're in a protected, climate-controlled container. You have to harden them off before putting them, in a different growing environment. And I think that covers our genetics and propagation part of the unit there on Chapter 9. Anybody have any questions, comments, concerns?
Yes, I had a question about one of the quizzes. Quiz 6, did you say Monday you had extended that, or did I hear you wrong about that?
Oh, yeah, I'm sorry. I might not have.
Yeah, it said it was due on Monday at midnight. That's what I was wondering. I tried to take it yesterday and it wouldn't let me do it.
Yeah, I'll open that back up to Friday for you. Sorry about that.
Thank you. That's okay.
Anybody else? All right. Well, you guys have a great day. We'll catch you next week. Remember to sign up for your crop projects on the discussion board.
There you go. Bye, everybody. See you in a few minutes.